Good afternoon and welcome everyone to this proteostasis webinar. I'd like to welcome Olivia Koo, one of the organisers of this seminar series, and our speaker today, Professor Alfred Goldberg, who will be talking on the functions of the proteasome from protein degradation to disease therapy. What we'd like to have your questions today at any point. If you have a particular question you'd like to ask, we'll have a short question session after the seminar. And if you have a question, part of the GoToWebinar function is that you're able to type your question out. If you could type your question, and then we'll come to you at the end. And Professor Goldberg's happy to answer questions that you have. So to start, I'd like to pass the seminar over to Olivier, who'll give you a brief introduction.
Okay, thank you, Sarah. Hello, everyone, and thanks for attending this webinar. That is the number 4 of the proteostasis webinars. So I am Olivier Cooks from Montpellier, France, and today it's a great pleasure and also an honor for me to introduce Fred Goldberg from the Department of Cell Biology of Harvard Medical School. I guess that most of you already know Fred Goldberg, so I will try to be brief in my introduction. And in fact, there is a very short way to introduce Fred, is to say that if we are all trying to understand the complicated processes of protein degradation, is basically Fred's fault, because he was the one who basically invented the field, or at least greatly contributed to open it. So Fred's career started at the end of the 60s, sorry, and he started his PhD by studying the control of muscle mass. And soon he became interested in the role of protein turnover in this process, and that was the beginning of a big scientific adventure, during which he remained focused on the two main issues of the beginning, that is the mechanisms of protein breakdown in both mammalian cells and bacterial cells, and the physiological regulation of protein degradation in normal and disease states, and particularly during muscle atrophy. So during this more than 40 years of research, Fred's contribution has been huge, and you all know that he is now recognized as the most important contributor and most influential scientist in the life science, as shown by the number of his publications and citations. So just in case some of you happen to not know Fred's career very well, I just want to summarize here some of his important discoveries. So first, Fred discovered the ATP-dependent proteases responsible for protein degradation both in bacteria and in mitochondria. And then he discovered that the same system existed, ATP-dependent proteolysis existed in mammalian cells, and he discovered what we call now the Ubiquitin-proteasome pathway. At the time, it was called the non-lysosomal ATP-dependent pathway for protein breakdown. And soon he shown also the involvement of the 20S and 26S proteasome in this process. And in fact, Fred is the one who invented the term proteasome. I was told that was during a ping pong match with Dr. Arrigo in Vermont during a meeting. Fred also investigated the role of the ubiquitin-proteasome pathway. He has shown that it was critical in the clearance of misfolded protein and in muscle atrophy that we can see in many disease states. And it also played a huge role in the dissection of the role of the ubiquitin-proteasome pathway in antigen presentation. And the last thing I'd like to point out is huge contribution in the development of proteasome inhibitors. I think that is very often underestimated, but you have to realize that before we had these inhibitors, the specific inhibitors of the proteasome, it wasn't clear that the proteasome was such an important enzyme in the cells. And only since Fred started to distribute these inhibitors to all the scientific community that we rapidly discovered that in fact we need a proteasome in almost every single biological pathway in the cell. So I think this contribution here is really very, very important for the recognition of the whole field and the importance of the field, particularly in the clinic, because you know that the inhibitor Bortezomib or Velcade is one of the major drugs against multiple myeloma now. So thanks to all his contribution, Fred was a recipient of many prizes. He was invited all over the world to give keynote lectures. Recently, he has been elected as American National Academy of Science, and so it's a huge honor for us to have him today to educate us about the proteasome. And just before finishing, I want to remind you that you could ask a question at the end of the seminar, and this webinar also will be recorded as a previous one, and you can watch all the webinar at the proteostasis on the proteostasis website. So Fred, now we will turn to you, and if you can take over the screen. And thank you for being here.
Whoops. Thank you very much for the kind introduction. Are my data still visible?
Yeah, but put it full screen.
I thought we had screen. I'm sorry for this difficulty.
That's okay, Fred. If you just press the full screen button that you've got it hovered over now.
I have
at the bottom. Yeah. No.
Yes. Is that okay now?
No. If you can press the ensure that you've pressed the Show My Screen button on the GoToWebinar. There we go.
I'm sorry for this difficulty.
That's okay. Yep, there we go. Yeah. Brilliant.
Good. Well, thank you very much, Olivier, for that wonderful, kind words and for reminding me how old I am. The lecture today, I will try building on the 3 previous talks that have emphasized how important is the problem of protein folding and homeostasis of protein structures. And now for over 40 years, as shown on the next slide, my slides are not changing. Yes, is that now, has the second slide appeared?
Yeah, we can see that second slide.
It's just lost for me. I don't know about these technical problems.
Okay, if you then...
Okay, here, it's okay here. Now, the second slide is on my screen. Is it on yours?
Yep, we can see that, no problem.
Fine. Over 40 years ago, we and others demonstrated the important role of protein breakdown in cells in eliminating misfolded proteins that we now know have escaped the chaperone mechanisms for protein folding correctly. In all cells and organelles where it's been examined, such proteins are rapidly degraded, as we found by an ATP-dependent process that does not involve the lysosomes, which at the time was the only known degradative system. By misfolded proteins, we know a large variety of mistakes in protein folding, post-synthetic damage can occur. In fact, the next slide, I'm sorry, the screen is not progressing. I am trying to get the second slide.
That's okay. If you just click on the screen that you're on and then try again. There we go.
Okay. This is a little bit of protein sociology that shows the kinds of proteins that are rapidly degraded in all cells. Incomplete proteins, as may arise by nonsense mutations or mistakes in RNA processing. About 20% of the missense proteins that most perturb protein folding. Excess free subunits that occur in multimeric complexes normally, but when they're free, tend to misfold and be rapidly degraded. And of course, post-synthetic damage to proteins is occurring all the time through intracellular denaturation and oxygen radicals. And such proteins normally don't accumulate because of rapid degradation. Often, genetic engineering efforts are frustrated by the rapid degradation of the scientist's favorite protein because the proteins are recognized as misfolded and rapidly degraded. In fact, we don't know how often proteins are misfolding in the cell because of their efficient degradation. Now, in trying to understand this process, almost 40 years ago, we were able to show that the rapid degradation of such proteins, as shown in the next slide, is rapidly hydrolyzed by an ATP-dependent soluble, that is non-lysosomal degradative system, that's present in first reticulocytes, but now we know in all eukaryotic and prokaryotic cells. This system, in the analysis of the unusual ATP requirement, has been the subject of intensive investigation ever since, and the classic work of Hershko and Cellular and Rose led to the discovery of ubiquitinations playing a role. And a few years later, Lloyd Waxman in our laboratory and the group under Rex Steiner were able to show that such ubiquitinated proteins are rapidly degraded by a structure we name the proteasome. Now, undergraduates, even high school students, learn about this pathway that we call now the ubiquitin-proteasome pathway, as shown in the next slide. Now, this summary slide simply emphasizes that there are two roles of ATP. We were focused on understanding the ATP requirement because it was so unusual thermodynamically. In other words, there was no known protease that required ATP until we found these complex enzymes in bacteria that were a new kind of protease. In mammalian cell, there's one such enzyme we named the 26S proteasome that utilizes ATP to function. But in addition, as Hershko, Rose, and Chicano discovered 35 years ago, there's another ATP-dependent step which provides selectivity to the process. In this step, as they discovered, proteins are modified by attachment of a small heat-stable protein, later called ubiquitin. And it targets the proteins for rapid degradation by the 26S proteasome. Now, the main point about this pathway is that it generates small peptides. It completely degrades proteins, in other words, to fragments, 99% of which live in the cell for a few seconds. Perhaps 10 seconds is the half-life maximum for such peptides. They are converted quickly to amino acids, which can be recycled. and a small fraction, maybe 1%, taken up by the endoplasmic reticulum and presented on the cell surface on MHC class I molecules. This means that the ubiquitin-proteasome pathway also allows the immune system to screen intracellular space for foreign peptides, for foreign proteins, as would occur upon viral infections or bacterial infections or cancers that generate abnormal proteins and thus an abnormal repertoire of peptides appearing on the cell surface. And this is a very important role of the pathway, often overlooked. But let's talk a bit about ubiquitination, since it's fundamental to all we are talking about. Hershko, Chikanover, and Rose first discovered this modification as an ATP-dependent step and showed that the ATP is required to chemically activate the ubiquitin, as shown in the first step here. The highly reactive ubiquitin thioester is then transferred to one of the cell's many ubiquitin conjugating enzymes. We have 30 to 40 different ones in our genome, and they provide some selectivity because different E2s carrying the ubiquitins function in the modification of different sets of proteins. This step then leads to the really exquisitely selective step in which a E3 recognizes the protein substrate and with a specific E2 ubiquitin pair is able to quickly processively modify the substrate by attaching a chain of ubiquitins to one or more sites on the protein. And this is an isopeptide bond attached to the lysine amino group, not the protein sequence itself normally. And when there are more than 4 ubiquitins, the protein is rapidly degraded by the proteasome. The point I want to emphasize here, and that I think all biologists should be aware of, is that there are in the genome well over 600 different ubiquitin ligases, which means that there are more ubiquitin ligases than even protein kinases that allows protein breakdown to selectively remove and destroy proteins under very specific physiological conditions. So far, we only know about the function of a fraction of those, and there are many opportunities for selective drug development. But let me return to the hero of our talk, the ubiquitin-proteasome complex is so named because of the 26S proteasome shown here in the next slide. This structure contains 60 subunits and is probably 2% of the weight of most of our cells. It consists of 2 main parts. The central part, shown here in yellow, is a hollow cylinder inside of which proteins are cut to small peptides and was first discovered as an isolated protease or particle before its main physiological role was discovered, which is as part of this large 26S complex. Ubiquitinated proteins bind to the 19S regulatory complex, which binds the ubiquitin chains and takes them apart so that the ubiquitin can be recycled in subsequent rounds of protein degradation. The protein is usually globular and has to be unwound, and that is a function of the 6 ATPases that form a ring, shown here in orange, and that cap the 20S proteasome. They translocate the proteins into the core proteasome, in which they are rapidly degraded. The main point here is that protein breakdown here is occurring not free in the cytosol, but within a subcompartment that isolates the potentially dangerous 6 proteolytic sites away from the cytosol. And all the selectivity of ubiquitination, it involves selecting which substrates will be bound and injected into the core proteasome for further degradation. I'm going to focus a little on the ATPases, which are basic to running this molecular machine. The proteasome evolved in archaea before eukaryotes appeared. And in archaea, there's a 20S proteasome particle that's homologous, although simpler in organization, than our proteasomes. For example, we have 7 different alpha subunits. Archaea have 1 alpha subunit, 7 copies in the outer rings. We have 7 different beta subunits in the central ring. Archaea have 1 type of beta subunit carrying the active sites, and degradation occurs in the central chamber formed by the beta subunits. But in both, there's an ATPase complex, a hexameric ATPase of the AAA ATP families. And prokaryotes, such as archaea and mycobacteria, but not other bacteria, myxobacteria, rhodococcus are a part of this family, tuberculosis a part of this family. They have proteasomes like archaea and a hexameric ATPase, but they lack ubiquitination. Our proteasomes contain this additional lid structure which binds the ubiquitinated proteins and catalyzes their deubiquitination. This step gave the proteasome exquisite specificity beyond what's present in the archaeal version. But these 6 ATPases are quite homologous, and studying their functions, we and others have uncovered 6 roles of ATP hydrolysis in protein breakdown. To summarize them, the first role of ATP is in binding irreversibly the ubiquitinated proteins. Then the ATPases are able to unfold the substrates because only an unfolded polypeptide can be translocated through a small opening in the alpha rings, which you have to permeate to enter the central degradative chamber. In fact, this is a gated entry place, and one of the roles of ATP, which we now understand, is to open this gate, which allows translocation into the destructive central chamber. We now have an almost atomic understanding of this gate opening process, and let me just share with you some of my favorite electron micrographs and mechanistic insights. Through the work of several postdoctoral fellows, Nadia Benarouche, but especially David Smith, we were able to show that ATP binding leads to an opening that is the entry site for substrates. The X-ray diffraction by Grohl and Huber had shown that the 20S eukaryotic proteasome is actually closed at each end, but then in collaboration with the Finley lab, they were able to hypothesize a gating function. And if one removes these N-termini of the alpha subunits, substrates could enter and be degraded. This structure is the active one. This is really a very low activity one, where substrates can only enter when there's accidental occasional opening. But when ATP, or a non-hydrolyzable ATP analog, binds to the larger ATPase complexes, then the gate opens and substrates can enter. When ATP is hydrolyze the ADP, the gate is closed. And in the intracellular environment where potassium is high, the proteasome is maintained in an inactive environment, even at free 20S, by gate closing. Now, the mystery of how it opened was advanced when David Smith noticed that in the archaeal ATPases, all 6 identical subunits have a hydrophobic tyrosine X motif. And a similar C-terminal motif is found on 3 of the ATP in eukaryotic proteasomes. And they were able to show that the binding of the ATPases involved the interaction of this motif with openings on the top of the proteasome alpha ring. In fact, this was documented by mutagenesis, by destructive enzymes, or most beautifully, by taking C-terminal peptides and adding them to the 20S proteasome, as we and also DiMartino's lab were able to do. This step, we now know, allows the C-terminal peptides to bind to these pockets found between the alpha rings. This is the X-ray diffraction image from 10 years earlier that we really didn't understand. And it became clear from our work and Bill's work that these pockets have a critical lysine residue that the ATPase's Hibix motif interacts with and cause gate opening. A talented cryo-EM expert, Yifan Cheng, collaborated with David Smith to obtain the following images of the archaeal proteasome. In green is the X-ray diffraction from the work 10 years earlier. The cryo-electron microscopic density, coming from 20,000 different images, is shown here in yellow. The red are where the peptides bind if they contain a Hibix motif. If that Hibix motif is destroyed or lacking, the peptides do not bind in this specific locus in the alpha ring openings. This is enough to open the subunits, allowing other peptides to come in and bind to the central beta rings where the substrates bind and are hydrolyzed. We now know much more about this structure, and from further electron micrographs, looking down on the 20S proteasome, where you can see an image as shown in the right here. The cryo-electron micrograph densities show the amino termini of each alpha subunits, and they cross in the middle in prokaryotes and touch each other in eukaryotes, making a barrier that substrates can't permeate. Interestingly, the earlier X-ray diffractions could not resolve these structures that the cryo-electron microscopy shows. Then when the peptides are added, as occurs with ATP binding of ATP, one sees a gate opening, gate closing, gate opening. I love these images because they are a near atomic resolution of the critical step leading to protein breakdown. The other ATP-dependent steps that allow unfolding and translocation are still not understood at this kind of near atomic resolution. Now, the 26S proteasome structure has been resolved much more beautifully in the last couple of years from the work of Baumeister's group and Martin's group. And we really are trying to understand how these different steps are coordinated. The substrates we now know bind to RPN 10 and 13, as was shown by Dickich and Finley's groups, which are found in the top of the proteasome. Then there's the deubiquitination by 3 different deubiquitinating enzymes that take apart the ubiquitin and recycle it. Otherwise, it can't enter through the ATPases. And RPN11, for example, is located right above the ATPases to remove the ubiquitin chain, allow it to enter, and be degraded. One important insight, we think, came from the work of Andreas Peth that shows that the proteasome itself lacks full activity until it binds to ubiquitin conjugates. In other words, the binding of an appropriate substrate activates the proteasome and stimulates multiple activities. We would like to understand all of them as well. And just to show you a bit of data, if you add a ubiquitin conjugate to the proteasome, you can activate many-fold the ability to hydrolyze a model peptide. Also, the critical step of hydrolyzing ATP is activated. So the proteasome is maintained in a relatively quiescent form until a conjugate binds. And the conjugate has to have both a ubiquitin chain and a loosely folded domain, as was shown by the work of Matushev. For example, if you add a ubiquitinated enzyme, dihydrofolate reductase, you activate ATPase. But if you add the strong inhibitor, methotrexate, an anti-cancer drug, that keeps the protein tightly folded, ATPase is kept at a basal level, the protein is not bound irreversibly and not degraded. So the proteasome is smarter than we thought. It's making decisions on requiring a ubiquitin chain and a loose end of the protein. To put our present mechanistic insights together, the ubiquitin chain first binds to the receptors, RPN 10 and 13, and the deubiquitination process begins. This itself triggers activation of the ATPases and gate opening. If there's a protein loosely folded someplace that the ATPases can attach onto, this allows the translocation of the substrate through the ATPases into the 20S proteasome and irreversible degradation. The first steps are reversible. Then there's a decision step which commits to degradation. So many ubiquitinated proteins are probably binding but never go on to irreversible degradation. Now, as Olivier mentioned, we were able to introduce about 20 years ago now inhibitors of the proteasome. And I think it's good to think about how drugs may be developed. The challenge is most of what I told you just now was completely unknown. The structure and the role of the 19S was vaguely understood. What we did know is the core 20S proteasome had 3 types of active sites that were cleaved after hydrophobic residues, basic residues, or acidic residues at different sites. And there are 3 such active sites in each beta ring, as we now know. What we did in setting out to try developing drugs against the proteasome was to try getting small molecules that would fool the proteasome, that would mimic these kinds of substrates. And we knew that it was possible to inhibit proteases, such as serine proteases, with aldehydes, which interact with the nucleophilic hydrophilic water-like residues, the hydroxyl group. We thought the protease was an unusual serine protease And the sequencing showed that it was a new kind of enzyme, a threonine protease, where the hydrophilic attack is by the hydroxyl group on the terminal threonine region, which allowed the development of very specific inhibitors. The first compounds we introduced were simple hydrophobic peptides, aldehydes, with a phenoxy group or another hydrophobic group to help the peptides get across membranes. This molecule, which has now been used in 11,000 scientific papers, probably by many of you in the audience, is simply 3 leucines in a row, and thus it resembles a high-affinity substrate for the chymotrypsin-like site, and it forms a transition state complex, shown here. Then in a small company, we founded called Myogenics, that's therefore the MG name. This molecule was converted by a team led by Julian Adams, first into a boronate, which was known to be a good serine protease inhibitor, and the boronate residue increased efficacy 50-fold, and within several months of medicinal chemistry, Julian and his team were able to generate this molecule, PS341, or Bortezomib, which has now been a drug taken by over 700,000 cancer patients around the world. This molecule does just what you'd expect a proteasome inhibitor to do. It inhibits certain key transcription factors like the production of NF-kappa B. Whoops, sorry. It stabilizes key cell cycle regulators. By itself, it can induce apoptosis because protein breakdown is essential. And it causes the accumulation of misfolded proteins, leading to ER stress and the heat shock response. Misfolded proteins in the endoplasmic reticulum and in the cytoplasm induce these responses, as you've heard. Originally, we were hoping to inhibit the excessive protein breakdown found in certain cancers, and the wasting of muscle and the atrophy of muscle. With time, it became clear that proteasome inhibitors were probably safest to use only in serious diseases, such as cancer, and it was eventually went into patients in phase 1 trials against all types of cancer. And dramatic effects were seen with a cancer of the plasma cells called multiple myeloma. This is an unusual cancer because it generates abnormal immunoglobulins in huge amounts. In fact, much of the disease is caused by the accumulation, in this patient, of a immunoglobulin A. This patient was on death's door. He resisted all of these drug treatments. Then when given in phase 1 trials, Bortezomib, the serum cleared of the abnormal immunoglobulin, which was destroying his kidneys, and the bone marrow got cleared of the myeloma cells. In other words, this has turned out to be an effective drug that was even approved by the FDA only after phase 2 trials because of its clear effects. There are now 3 different proteasome inhibitors that are approved for human use around the world, and they have now, in combined with other drugs, over 95% of the patients are showing a clear response. That has lengthened the lifespan. Now, this has been the great majority. This has been very gratifying, but the truth of the matter is that the special relevance to this disease was only discovered in subsequent years. And the reason why it's so uniquely effective in this disease and not other cancers is because this is a disease dominated by the production of an abnormal protein. This is really a proteostasis disease. The immunoglobulins are very difficult to synthesize, and a large fraction of them are normally removed by the quality control ubiquitin-proteasome pathway, specifically the branch called ERAD that pulls the abnormal proteins out of the ER and targets them for rapid degradation. When you block in the cytosol by the proteasome, when you block this pathway, you further cause activation of the unfolded protein response, and you trigger programmed cell death because cells are overwhelmed by misfolded proteins. Now, this is very toxic effect, but myeloma cells, like all cells, need NF-kappa B to inhibit apoptosis, and the production of NF-kappa B is also blocked by these drugs. And white cells, and in particular myeloma cells, like the normal plasma cells, really require NF-kappa B to produce growth factors like IL-6 or factors that are necessary for the destruction of bone where this cancer grows. So this combination of effects has made it particularly effective against this disease. Now you may ask why, since the proteasome is so important, do these treatments not kill every cell in the body, which was the anticipated effect? And sure enough, the explanation is quite simple. These approved inhibitors mainly, and at most doses exclusively, block the chymotrypsin-like site, which means that proteins are still degraded by other sites of the body, just at 60% of 50%, maybe 70% normal rates. Cells can certainly compensate. The chymotrypsin-like site is absolutely essential for the huge amount of protein breakdown that the myeloma cell has to perform. So these cells are particularly sensitive and go into apoptosis. Cell biologists use these drugs at much higher doses, so they have more of an effect and also inhibit the caspase-like site. But still, some protein breakdown occurs by the remaining trypsin-like site at a slow rate. There's also, we now know that cells can compensate for the inhibition of the proteasome by the induction of more proteasomes by a specific transcriptional response, and to some extent by activating autophagy, and we are now working on the mechanisms of these adaptations. Because we started a few minutes late, I'm going to skip our knowledge of these mechanisms and recommend that you might want to look at a recent paper by Sharon, myself, in Current Biology that has described, we think, the novel mechanisms by which new proteasomes are made by cells when they lose proteasome function. And I'd like to end with a very quick summary of what I think is the future of the field, a very exciting development that I'm going to skim over. And this is the recent discovery that proteasome function can be regulated also by phosphorylation. In our case, by phosphorylation by the protein kinase that's activated by cyclic AMP, pKa, that enhances the proteostasis capacity. This very weak phosphorylation of the proteasome of a different kind by a different kinase has been shown to be important in the cell cycle by Dixon's group. So this is going to be an emerging area, a new form of regulation of protein degradation that you're going to hear a great deal about, mainly because this process, I think, has therapeutic potential against a whole other group of diseases. And in 3 or 4 minutes, I'm going to give you a Reader's Digest summary of these developments and recommend that you, if you're interested, read these papers. The cyclic AMP pathway is probably the best known pathway for signal transduction. In fact, a couple Nobel Prizes were given for understanding its role, and it mediates many hormone and neurotransmitter actions. What we have found, it was suggested earlier by others, that you can activate the 26S proteasome against peptide substrates and ubiquitin conjugate degradation by stimulating the accumulation of cyclic AMP. In this case, with a drug called Rolopram that blocks the hydrolysis of cyclic AMP by phosphodiesterase 4. You can mimic this in vivo effect with a pure protein kinase, and you can reverse it with a pure protein phosphatase, PPI1, which modifies selectively RPN6, one of the regular subunits on the top of the proteasome. This occurs in vivo with Rolopram. It occurs with a pure enzyme, PKA, and it can be blocked with inhibitors of PKA. And sure enough, that modification alone genetically mimics the activation. If you use a mutation that mimics the phosphorylation of RPN6, you activate the proteasome, and you can inhibit it if you prevent this phosphorylation. So this raises the question, what happens if you activate the proteasome? Do you affect all protein degradation, or might you affect some? And I'm mentioning it in a course on proteostasis because we found that it selectively blocks the breakdown of misfolded proteins. Specifically, recently synthesized short-lived proteins are degraded faster, but not the great bulk of proteins, which are long-lived components. And activating protein kinase promotes the degradation, in this case, of a mutant protein, SOD1, that is responsible for a form of hereditary ALS, amyotrophic lateral sclerosis. And you get this accelerated degradation even if you block lysosomes with concatamycin A, but not, it's blocked if you block the proteasome or ubiquitination. And you can ask if such an agent, such a mechanism, can affect the clearance of toxic proteins in the brain. And in collaboration with Natura Mayeko and Karen Duff, we have recently shown that in a model of a disease resembling Alzheimer's disease, an inherited mouse model for frontotemporal dementia, in the mouse you see ubiquitin conjugates rising in the brain and the toxic protein phosphatase rising, while 26S activity goes down. In the crude extract, and when you isolate the proteasomes, you find with time in the disease a reduced ability to degrade ubiquitinated model proteins and reduced proteasome activity in the brain, specifically of the 26S, as ubiquitin conjugates accumulate. And in the final slide, if you treat these mice with roloprand, which raises cyclic AMP and activates proteasomes in the brain, the levels of this toxic protein go down. So we have exciting data now to indicate a possible route to treat neurodegenerative diseases and other proteotoxic diseases where there's an accumulation of misfolded proteins. But the main point to end in is really true. The proteasome, we think, is really an exciting, interesting structure, even more interesting than many of us thought. It has exquisite endogenous regulation. It gets turned on, that is ATP hydrolysis, gets turned on when you bind a ubiquitin conjugate, which interact with DUBs and induce major structural changes, as shown by the recent work of Baumeister and Martin, that activates the particle. And then there's also exogenous activation. Our work focused on protein kinase A that hydrolyzes misfolded proteins. And so regulation is not just at the ubiquitination step, but also at the proteasome through phosphorylation and probably a number of other types of modifications. Thank you, and I'm sorry for the technical difficulties and that we have little time left. But I'd love to answer any questions you may have or that I may have caused.
Thank you very much, Professor Goldberg. That was a great talk and very insightful. We do have a few minutes left, so if you do have any questions, please, if you could type them in the question function of the GoToWebinar. We do have one already up, and that's from Phanindra Kumar Deshmukh from the UK. I'm going to attempt to unmute Phanindra. Apologies if I've pronounced your name incorrectly, and we'll see if we can get you talking in person. So I'll just unmute you. Are you there, Phanindra?
Yeah, I'm here. Thank you for taking this question. I didn't thought that I would asking this question direct to the Professor Alfred. So my question is that it was very interesting talk, and my question is that whether proteasome has two entry sites, or is it any specific? Is there any specific site on the proteasome that only through one side a protein enters, and only through one side a protein exit, or it is a two-way entry?
I mean, this is an important question that there's no question that the 26S proteasome comes, whenever we look, in two forms. One form that looks symmetric with a 19S regulatory complex at both ends, and one form that's quite abundant also that has such a complex at one end, which has to be the one that is active. At the other end of those complexes, there are additional PA200 11S regulatory complexes. So it looks like in those singly capped forms, there is a polarized one-direction entrance. In the doubly capped form, which most of us study, we do not yet have a way to ask whether substrates enter from both ends or only from one end. This is a biophysical challenge to approach. It may well be that a conjugate can act at one end. We draw it with substrates coming one end and products going out the other. That's just for simplicity and to make teaching easy. Products could still be exiting through the same entry point in the alpha ring that the protein originally entered. So I can only say that this is a good question and wish I knew the answer.
Thank you very much.
Thank you, Professor.
Thank you, Phanindra. We have another question from Naveen Kondru. So let me just unmute you, Naveen. And are you there, Naveen?
Yep.
Go ahead and ask your question.
Thank you. Dr. Goldberg, it's an interesting talk and I enjoyed it. So my question is, so if we inhibit a proteasome in cancer therapy, so you're going towards a clinic, so does it lead to an off-target effects like accumulation of misfolded proteins and resulting in other protein misfolding disorders like tau pathology and A beta pathology like that?
Yeah. This is a important question which really concerned us in the early days. First of all, let me emphasize we are not going to the clinic. This has been an approved drug for 11 years now and has been taken by 700,000 patients. So the answer is we are not causing pathology in the CNS because, as was established very early, these drugs do not cross the blood-brain barrier, and thus we do not get any accumulation of toxic proteins of the kind that you are raising. In cells, however, this is a very good tool to cause the accumulation of mutant proteins or to study these diseases. Yes, there are side effects. They're not very well understood. Some peripheral neuropathy that you see in some patients may arise by accumulation of misfolded proteins. But as I emphasized, at these doses, one is only partially inhibiting protein breakdown. And cells seem not to be affected, but these particular myeloma cells are so overwhelmed by misfolded proteins that they alone go into apoptosis. Normal plasma cells are also sensitive, and this agent can be used and is being studied as an anti-inflammatory agent to treat lupus and to treat certain other immune diseases.
Okay, thank you.
Hello?
Sorry, hi. So we're rapidly running out of time at the moment, but there are a fair few other questions that have come in from people who've been watching the webinar. So we will pass those on to you, Professor Goldberg, and see if we can get any answers to the questions that people have been asking, and we'll see if we can get those out to all the people who've participated today. So I'd like to thank everybody for joining us today, and I'd especially like to thank Olivier and Professor Goldberg for their time and their insights into this very exciting area. So thank you very much, and have a great day wherever you are in the world. Bye-bye.
Bye-bye.